This is the last topic for the second day of this course. We have seen the examples of nucleotide databases in the previous topic. We shall now look into a specific protein database throughout this lecture. Let's begin. One of the most important protein databases came from the collaborative project between three different organizations that hold different types of protein databases. These organizations are the Swiss Institute of Bioinformatics, the EMBL EBI, which we have briefly discussed about in the previous lecture, and the Protein Information Resources databases. The result from this collaboration was the unification of protein information and the development of the UniProt database in December 2003. The UniProt comprises of three different databases which are optimized for different users. The UniProt knowledge base, also known as UniProt KB, is used to access functional information on proteins. A subset of the UniProt KB entries form the proteomes dataset. This consists of the set of proteins thought to be expressed by an organism whose genome has been completely sequenced. Meanwhile, the UniProt reference cluster, also known as UniRef, provides cluster sets of sequences at several resolutions. The UniProt archive, also known as UniPark, is a sequence archive which contains all protein sequences from the main publicly available protein sequence databases. UniProt contains high-quality, manually annotated and non-redundant protein sequence records. Manual annotation consists of a critical review of predicted data for each protein as well as manual verification of each protein sequence. UniProt curators extract biological information from the literature and perform numerous computational analyses. It aims to provide all known relevant information about the particular protein. Annotation captured from the scientific literature includes information on protein and gene names, function, catalytic activity, cofactors, subcellular location, protein-protein interactions, and much more. The UniProt SwissProt contains manually created highly annotated sequences from SwissProt, including descriptions, taxonomy, citation, geoterms, motifs, functional and structural classification, residue-specific annotations, including variations. Some automatic rule-based annotations include interpro domains and motifs, prosets, prints, prodom, smart, pfam, superfamily, and tigerfarms classifications. While the UniProt Chumble is a computer-annotated database which contains automatically translated sequences from genomes, including predicted as well as RefSeq genes, and automated rule-based annotations. Under the UniRef, there are three non-redundant collections based on sequence similarity clusters. The first one is UniRef 100, which has all identical and identical overlapping subsequences merged into one entry in UniRef 100. UniRef 90 merges all protein sequence clusters with 90% sequence identity into a single entry. Meanwhile, under UniRef 50, merged all sequences clusters with 50% sequence identity into a single entry. Under the UniProt archive, also known as UniPark, the database contains stable, comprehensive, non-redundant collection of all protein sequences ever published. The UniRef databases provides cluster sets of sequences from UniProt knowledge base, including splice variations and isoforms, and selected UniPark records in order to obtain complete coverage of sequence space at several resolutions. UniRef 90 and UniRef 50 are built by clustered UniRef 100 sequences with 11 or more residues using the CDHIT algorithm, such that each cluster is composed of sequences that have at least 90% or 50% sequence identity, respectively, to the representative sequence. The UniProt archive is a comprehensive repository reflecting the history of all protein sequences. You may search for the UniProt database on any online search engine like google.com, and you will see the web interface just like the one on the screen right now. UniProt is a comprehensive repository of protein sequences and their functional annotations. Curators add value to raw data by annotations against scientific literature. The main purpose of UniProt is the creation and maintenance of stable, comprehensive, and high-quality protein database with high-level accessibility to facilitate cross-database information retrieval. In this topic, you have learned about the brief introduction about UniProts, the three major components of UniProt knowledge base, which are the UniProt, UniPark, and UniRef. The relationship between UniProt, UniPark, and UniRef. Thank you for your attention.